Thank you. Hey, I'm Xuefang Zhao. I'm a second-year PhD student, now working in Dr. Ramel's lab. So today I'm going to talk about the algorithm our lab have been working on in the past year, which is called the SVelter. Yeah, which is called SVelter. It is a randomized approach used to solve a complex structural variance. So in this presentation, I'm going to talk about the following questions. First, we're going to define the structure variance and complex structure variance and talk about why we're spending all this time studying them. And second, we're going to talk about the method we developed, like how does SVelter try to identify and also resolve SV and CSV. And then we're going to talk about the current result we have and what more we can expect from the method. First, let's define structural variance. By definition, structural variance are the rearrangement of DNA sequences that are usually larger than 50 base. If it's smaller, we usually call them SNP or indels. So they're usually caused by deletion, duplication, inversion, insertion, or translocation. For each of them, I have a example in the next slide. And structural variance is sort of very important because first they play roles in a lot of cancers as well as other very severe syndromes like this Prader-Willi syndrome, which there is a very long deletion on chromosome 15, which causes something I don't know how to describe. Sorry. So also smaller structural variants that happen on smaller region are also very important because they serve as a way for they play roles in evolution, in population differentiation, and also for disease phenotypes. So here is the 5 basic type of structural variants, where the gray bar stands for sample 2, while the black bar stands for sample 1. So compared to sample 2, for example, here there is a deletion, so there should be a DNA material here on sample 2, but in sample 1, this part got deleted or something. For duplication, there is one red bar here on sample 2, while in sample 1, this part got duplicated and inserted somewhere else. Maybe close to the original copy, but it may also be very far in some other chromosomes maybe. So this definition is sort of relative, based on one sample compared to the other. But in reality, we really do need some uniform format of sample 2, which we use the human reference genome. So when we define the structural variance, we were talking about is the genome structure of the sample we have compared to the human reference, AG19 to be specific. So as SV, it's important and interesting. So there's a lot of method already there to study structural variance. We got DALI, we got Meerkat here, which works pretty well, and also we got breakdancer, SVelter. All those methods has been published before. They work pretty well, I think, in simple structural variants like one-step deletion or one-step inversion, things like this. But in reality, the situation is really not this simple. For example, here are 2 cases. This is the genome structure from a germline genome in one person. So we can see there is a mosaic structure between chromosome X and chromosome 5. So let's see, there are 9, 10, 11 different regions from the 2 chromosomes that are patched together and inserted here. And also there's a very long material from chromosome X that got deleted from X and then inserted on chromosome 5, and here again part of materials from 5 are in chromosome X. Although this is more complex in second person, where 3 different chromosomes are involved, and it's too messy for me to describe.
When you look in the population or in that specific family, like did all of those happen in one person, or was there like is there a history of this kind of complex?
For
There's a lot
that.
of events. The whole set.
Well, these are two separate individuals. But
No, no, no.
The whole shattering occurred in de novo parents.
Okay. So like this is for germline genome, but when it comes to the cancer genome, it is just more complex. So here is a chordoma sample, where we can see like 5 different chromosomes are involved here, and the purple ones stand for the DNA materials that are interchanged between the 2 chromosomes on both ends of the purple line. And also there's like yellow one stands for the inversion. The green one stands. Oh yeah, there's also head-to-head and toe-to-tail. Various types of structural variants here, and they are all messed up together. So before we really talk about like how to solve this sort of situation, let's first define the.
There's certain regions that are more prone to these events. Some of it is concentrated in.
So we didn't really. That's sort of our next step to check for this. But as far as we can see, of course, like the centromere and telomere regions are really messy, so we were not able to study for this. But for the other regions, I think they're handleable for us. So complex structural variants are defined as complex rearrangement on the chromosome that are resulting from either multi-stop or overlapping events that involves at least 3 breakpoints. So they are important as they play roles in a lot of tumors and congenital or developmental defects. So I'm going to give examples of how complex structural variants look like. So in this case, it just happened on 1 chromosome. So there's 2 regions on this chromosome, the purple one and also the red one. So here, what happened is like the purple one got duplicated, and one of the duplication got inserted, and the red one just got deleted from the part, and all these others are like duplicated normally. So this is like the structure of the 2 homologous chromosomes in 1 person, where one chromosome look like this while the other look normal. By normal, we mean it's the same with reference genome. Here is a example of the overlapping complex structural variants, where we have very different structural variants happened on both chromosomes, but they happen on the same region on a pair of homologous chromosomes, but the rearrangement there is different. Like for 1 chromosome, there is a very long deletion, while for the second, there is a smaller deletion with a smaller inversion behind it. So both of those are like real case in our study. I'm gonna show like the real case later. So to solve this like situations like this current approaches we have that have been published have their limitations, because most of this method, like when they start to solve this sort of problems, they already set some expected patterns of particular variance types, and they're trying to search for this variance, this very abnormal reads that could match their expectation. But the problem here is, so there's various different types of variants we can have in the genome, and it's really hard for one person to imagine all the possible situations, right? So that's why the current approaches have some problem solving the complex examples I've given before. Let's see, this is a real case on chromosome 2, which is sort of popular in population. This is how we've been doing here. So this is like what should have happened as we talked before. This purple bar got duplicated and inverted while this got deleted. Well, for the current populations we have, there is 2 studies that have detected there is an inversion on this region, but they didn't detect there is like a duplication and insertion somewhere else. And none of these studies have detected there is another deletion very close to this part. So here comes SVelter, which is the method we've been working on. So as the input data, SVelter would take per end the next generation sequencing data. So with the initial BIM file, we would first build the node model based on the repair insertions, the redepths, and also the split read. Also, we would consider the direction of a pair of reads, which I'm going to explain more later. So the output of SVelter, which is I think is pretty cool, is we will output the resolved whole genome structure. So as the best structure we output, we define them as the one with minimized aberrant read mappings. So here's a very brief introduction about next generation sequencing. So when we do the experiment, we share the whole genome into pieces that are within certain lengths, and then we sequence the both end of the sequence, the DNA pieces we have, and here they could probably produce a FASTQ file for us. And with this file, what we do is we map each pair to the reference genome. So theoretically, if there's like no structural variance on the sample we have, they should map back very beautiful like this, right? Like there is, I'm going to talk about it here. So for all the read, their insert lengths should within some distribution as how the technique said it. We fit the distribution here as a mixed Gaussian distribution. And also for like each pair, we would expect their direction to be forward and reverse, because when we sequence, when we do the sequencing, we just sequence the both ends in the same direction, right? So there are 3 different situations like this. They are all wrong structures, and they are telling us there is probably some structural variance happened where this reads mapped. Also, we would try to model the redepths based on the negative binomial distribution, because when we do the sequencing, we just randomly choose like a sequence from the sample pool to get sequence. So theoretically, they should follow like a Poisson or negative binomial distribution. But we found that this model could also be fit very well by normal distribution. So like the users, I mean, someday when the method is publicly available, the users could choose either negative binomial or normal distribution to fit their redepth model. So here it comes to how the SVelter really solve complex structural variants. Here is a very brief workflow of all the thing, and I'm gonna talk about details later. So what SVelter do is we first define breakpoint, where we think maybe like the 2 regions on both sides of breakpoint could be different from the reference genome. And after these, we define the blocks based on the breakpoint. Here we have 4 breakpoints here, so we can define 3 different blocks, 3 different potentially interesting blocks here in between. And also we will add 2 anchor on both sides, which is very important here. And then after we define the breakpoint, we have this sort of structure. We would randomly propose a structural variance, a simple structural variance, like deletion or inversion. Here, in this case, we propose a deletion. So we would delete all possible blocks. Like here, we delete A, we delete then B, and then C for this, and from the new structure. And for each structure, we will map all the reads back to the new structure, and we will score each structure like this. Also, we will score the original structure. So when we get the sort of score for each structure, we will transfer them into a probability model. And then we will choose a best structure from this 4 based on their score. But so usually, the higher a structure is scored, the more possibility it is chosen for the next step. But that's not necessarily all the cases. So after this, we will renew the structure to the picked one, and then we start a next round. Here's the brief workflow, just in Word. I think no one's interested, right? So first, let's talk how did we define the breakpoint. So we defined the breakpoint based on the signals from aberrant read pairs. So let's first define the aberrant breakpoints. By aberrant, we mean first if for a pair of read, if their insert length is very abnormal, if it is outside our expected distribution, either too large on the right tail or too small on the left tail, we would mark them as some potential sign. And also the direction, like in this case, for this pair, they are forward, while for this pair, they're reverse. Or we could also have like backward and reverse situation, which is not good. So we also mark these. And also clip reads means for a read, only part of the read got mapped in this region, while the other part got clipped and it mapped somewhere else we don't know. So we also take this as a very strong sign of breakpoint. After we define all these aberrant reads like this, see, if there's like a cluster of aberrant read, and they're clustered together means they come from the same read pair, we could define a pair of breakpoint. And the breakpoint has to be in front of the rightmost forward read if all the reads in the cluster are heading forward. And they also have to be on the very end of the leftmost read in the if a cluster is all consist of the reverse read. Am I confusing anyone? Also, if we have a clip read, or in that situation, if we have several different clip reads that are all clipped in the same position, we could also define a breakpoint there. After defining the breakpoint, our next step is to cluster different breakpoints together because so in our method is not capable of running the whole genome, or we are not even capable of running one chromosome at one time because there could be like hundreds or even thousands breakpoints on the chromosome. And there are so many different possibilities that the structure is rearranged. So if we just run them in one algorithm, it'll take forever to run. So our task here is we would try to define the possible interesting regions, or we can think it as a SV hotspot, where they have a cluster of breakpoint defined together. And we just focus on this local region, and we solve this local region first before we integrate all the information from other local regions. So here is a case like how it looks on IGV, where you can set whatever color you like. But here, the colorful stuff stands for like clip reads, while here the blue stands for the forward, and this is green, sorry. Green stands for the forward read, while the blue stands for the reverse read. Does anyone have problems with IGV screen? Okay, cool. Oh, also here it is telling you the coverage. So next step is to link different breakpoints.
Do you have a created set of known breakpoints? You're a car like this in a cluster. Do you have a known set of breakpoints that are well allocated that you can test against?
Yes, this is one of the cases. Yes, this is the chromosome 2 case, which is sort of very popular in population, and we can observe this in maybe a lot of people here. Yes, so this is a very interesting case we have for breakpoint linked together exactly in this format. We're going to talk more about this case later. So next step, we are going to link different breakpoints to form a cluster. By linking, we have a lot of different criteria like for this. If two pairs are overlapping, by overlapping we mean, so for example, these two breakpoints are defined by a cluster of pairs, while these two are defined like this. While the region are overlapping, so we would cluster them four together, and we think there could be something happened with those four all involved. And also, like situation like this is also defined as overlapping, or like if two breakpoint defined are too close to each other, maybe smaller one than 1 kilobase, we would cluster these two and together with their linked breakpoint as well. And there's another situation which is more interesting and complex. We call fault travelers, where let's see you have a cluster of array pairs, and one of the read got mapped here, while the mate of this read got mapped somewhere very far, like maybe mapped in some other chromosomes or in mitochondria or very far end of the same chromosome. In this situation, we cluster this four breakpoint together, and we also would drag that breakpoint here, and we solve this structure together. Does anyone have any questions? So after we define all the potential interesting regions with all the breakpoints defined, we're going to be able to start a iteration step, which we propose the rearrangement. We virtually rearrange them and map the reads back, and we score, and we start a new iteration. I'm going to talk about it more here. So in the very initial step, we have a lot of reads mapped back, right? Among all these reads, we have normal reads, like the gray bars, and also we have abnormal reads like this. And first, what we do is with the reference sequence and all the aberrant reads, we score the structure first. And we define the breakpoint as we described before. And here we have the three different blocks together with the anchors. And let's see, we random propose our first rearrangement, which is a deletion. So we will delete block A first, and then we map all this read back. So this read could actually look like this. For the reverse blue pairs, non-parameters in them are changed. While for the green ones, like their insert length is much shorter because of the loss of A. So if we delete B, again, this insert length of the green pairs would be shorter. They will be closer to each other. But for the other pair, we got a problem here, right? So like their mate, so this reads are mapped perfectly fine. That's good. But for this, they're mapped nowhere, which indicating we're not doing this right, which tells us this is not a good structure. So we probably give a very bad score for this structure. And also we could delete C. And in this case, we got 4 different reads that got not mapped anywhere. Then we also get a very bad score. After we got the score, we will transfer them to a probability model, which so we would first normalize the score into some certain range, and then we will add all the 4 possibility to 1, and we will randomly choose a probability between 1 and 2, and whichever block defined by the scores our chosen point fall into, we will take the structure as the right one we decide in this step, and then we will renew our structure like this. So here we choose the best score structure, and we renew the structure we have, and then we will start the next iteration. So in the next step, again, we will score the structure based on their current mapping situation of all the reads. And let's see, we random propose the second move called insertion. So insertion is a little bit different from deletion and inversion, because let's see, we could insert any possible blocks that are here, and also we could insert some far travelers that are linked from our former step. You still remember, right? So maybe D is defined by the breakpoint that are here, while their mate are linked somewhere else. Let's see here. And next step, we will choose a certain block here to be inserted. So here we choose C, and let's see how C could be inserted. If we insert C here on the first breakpoint we have, all the read will be remapped like this. So here, for this read, it won't be changed, and also this. So it looks, nothing have changed except for that the insert length for the green pairs are just longer, but it'll produce definitely a better score. And also the situation here is, so we can, for this pair, we can map them onto the C block here, right? But we can also map them to the block C here. They're the same spot. So in this situation, our principle is we choose the one that with the best static probability. So based on the insert length model we built before, this could be like maybe too long for us, so it'll produce a very bad score for this structure. But if we map the pair here like this, the insert length could make more sense for us, and this could end in a much better score, so we just keep this as a mapping situation. We decide all this in our algorithm. And also we could insert C here or here, but they're all the same, right? Like they would all ended up in this structure. So we just do it once. And we map all this read back. Again, we have two situations to map them, and we will choose the better one. And after we got the scores, again, we do the probability thing, and we choose the best structure. Here, I have to mention this in the part is we decided the best structure based on their score, but it's not necessary that the best score structure is always going to be chosen. Because if we really just choose the best structure, we will probably be trapped in the local minimum. This way, it gets us a very slight chance to like jump out of the local minimum and try to find the global minimum. So then we renew the structure, we add feedback here, and we start a new run. Let's see, we want to do an inversion here. So for inversion, we just could invert the first block, the second block, or the third block. And we will check one by one how their reads are mapped. We can see here, this is obviously a much better case because let's see, the direction are all correct. Well, for this, the direction are wrong. So maybe this structure shouldn't be scored very good, but we have other parameters like maybe in this structure, their coverage are become very bad, or their maybe insert length become very bad here. Anyway, this is the score we got, and then we choose a score here based on the score. Here, this is the situation where not the best structure is not chosen well, maybe the worst got chosen here. And then we renew the structure. And we remap all this and we start a new iteration again and again. So these are three basic structure variance situation. So we have two more. We have duplication and translocation. But we found that duplication, translocation could be concluded by the three, because think about it, if it's a duplication, we can replace it as an inversion, right? I'm not gonna go back, it's too long. So if it's a duplication, maybe if we want to duplicate B, we can just insert B back somewhere. And this is like a duplication. And also translocation, we can delete first and then insert back somewhere else. So I guess we all have the question is like, how do we come from the mapping of all the three to the score of each structure here. So our score system actually are composed of 4 different factors. The first is the distribution of insert length. So we will calculate the posterior Bayes probability of each pair based on this distribution. For example, let's compare between these 2 structures. Here we have the insert length of this pair, while here we have the insert length 2 of this pair. It turned out that insert length 1 located here is larger than like 600 base, and the probability of observing such a insert length is very low in this distribution. While if we go to insert length 2, the probability is much better, which causes a better insert length score for this structure compared to this one. Also, like for the direction, let's compare this to situation with C inverted or not inverted. So in this situation, we have 4 aberrant read pairs that are forward, forward, and reverse, reverse. Thus, we have four pairs with all wrong direction. So the penalty for direction here is 4, while the penalty, if we invert C, we will have a 0 penalty for the direction. So the decrease in the penalty score would also cause a much better score for the structure. Again, we also consider the coverage and the number of repairs going through each breakpoint. Let's see here. It seems that A got deleted because it gets a much lower coverage, while C got duplicated because it gets a higher coverage. Okay, here's a systematically format how we are scoring the structure. We got insert length, direction, read depth, and the number of reads going through a breakpoint. Both the direction and the breakpoint, these 2 are served as an actual loading factor for the insert length and read depth. So we first calculate the posterior Bayes probability of the insert length based on the mixed Gaussian distribution, and we take the log of it. And then for the directions penalty, we will calculate the percentage of reads that got abnormal directions, and we time the log PL with 1 plus the percentage here. And that's from our repair score. And also here we calculate again the posterior Bayes probability of the read depth based on the negative binomial distribution or normal, whatever you like. And we take the log of the probability, and we add this as an actual loading on the score, and we form read depth scoring. And then we will add them up by giving them different weight, and we form the final structure score, which is what we show. Sorry, it's too long back. This is just the final score we're gonna give for each structure. So far, I sort of have been done talking about the whole workflow we've been working on, but we're sort of naive because we need to respect the fact that human got diploid genome. So by only solving like 1 chromosome is not enough. We have to consider the pair of homologous chromosomes together when we do the scoring, because so for each read pair we got, they could be either mapped on the chromosome you get from your mommy, or they could also be mapped to the chromosome you get from your daddy, right? So when we are manually mapping them back, we have several principles we have to follow. First, if the 2 structures are the same, of course, we will try to distribute the repairs as evenly as we can, because like in the real technique, you can pick them from anyone you like. But if the structure are different on the pair of homologous chromosomes, we are gonna make some choices. For example, if we have a pair of reads that are like this, they are mapped not very good on this allele. But if we map them to the other allele, they get a better score. So in this situation, we will choose this pair to be mapped on this structure. Based on this precondition, then we could do some modification to maybe take some read from here, map back to this structure to make the coverage look better. So in this way, we are gonna trying to find the best score for each structure. So here's how we actually do this. So for a pair of homologous chromosomes, we first propose a random move on one allele, and we do the we try the possibilities, and we choose the best structure, and then we would propose a second move on the other allele that has not been changed in the former step, and then we change the structure. And we renew the structure on both chromosomes. And we start the next run. So here's our final. So usually, the problem with the method we are now currently have is, so after a lot of iterations, our result just doesn't converge to the best score. So currently, what we do is we would run the first 100,000 iterations, and we will pick the structure with the best score. So like each iteration, you pick a structure. This structure may be not the structure may not be the one that are scored best among its own pool, but when we take it out, we will record its score, and after a 100,000 iteration run, we will pick the best score structure among this pool. But there's a situation if there is one structure that are scored as the best for continuously for over 500 iterations, we could consider it as falling in the global minimum, and we just stop the iterations and output this structure as the final one. Like for example, because our algorithm do not only aim to solve the complex structure variance, we would also like to solve the very simple structure variance, like a simple deletion inversion in this whole like workflow. For example, if we have just a block B that's interested in our local region, and in our first step, we just delete B, heterozygous maybe, and that arrives the right structure, and that kept being the right structure for over 500 times. So in this situation, it is meaningless for us to run like 100,000 iterations. We can just keep the result we have and output it as a final structure we want. So here I'm going to show some primary results from SVelter we currently have. This is the real case in chromosome 2, which we showed before. And this is a pedigree, grandparents, parents, and kids. So here in this sample, they have a heterozygous structure variance on one chromosome, while in this sample, they have a homozygous. This person have a homozygous structure variance, while their parents both have different sort of structure variance, except for this one. It has normal on both alleles. And when we look at their kids, this is the result we got. So several kids are homozygous variants, while some are heterozygous, and there is no normal kids here. And this, the pedigree follows the Mendelian principles, even though we haven't tried to validate all the structures in the real lab. Also, we tried the bigger challenge we have here is the overlapping events we have on chromosome 3. This is the overlap like with a long deletion with a smaller deletion inversion on the other chromosome. And here's the result we have. So for this sample, the long deletion happened on both alleles, while for this sample, the overlapping events happened together with this. And in this family, they're all normal. Their structure are like what's in the reference genome. And we can see in their kids, there are several, all their kids have one normal chromosome, while for the other, some get the bigger deletion or some got the more complex one. And also this pedigree follows the Mendelian principles, which we think is recurrent. And for this special case, we also went back to look at the PacBio data with longer read. And that data also support our structure at least in the sample NA12878. Well, that's all it. And I would like to thank my boss and all the people in my lab. Thank you. Any questions? Oh, okay. So for this case, a 10,000 iteration usually takes like half a day to a day. Yeah, it's to solve one local region. But like if you have enough cores, you can run all these regions parallelly. And so, I mean, for the iteration step, the whole chromosome are expected to get run maybe in one day if you have enough core. And also we're working on this to make it faster. And the breakpoint searching step, it's really fast. The breakpoint searching step takes like 1 or 2 hours. So and there is another step, like when we got all the read and we try to fit a null model based on the distribution of the insert length and read depth. That step takes like half a day because it has to run the whole BAM file to get all the parameters, like what we usually like use JTK for. So in total, this method currently takes 2 or 3 days.
So do you have an idea of why the speed is very low? It's because the model space is very large.
So for the no model setting step, it is slow because the BAM file is large. The BAM file is like 100 gigabyte, and you have to go through each read inside, and you have to see whether the insert length is normal, is within your expectation, and you have to calculate whether the coverage is within your expectation, right? So it takes time to go through the whole BAM file. And also, like, you have to know the higher coverage we have, the better result we could probably get. But the larger the BAM file there is going to be, so the longer it takes. For the iteration step, so I think it takes long because in each step, we have to remap all the reads back. And it's not just the abnormal reads. Think about it, if you change a structure, there are some reads that are originally like normal mapped, right? They could be abnormal. So if there is, like in this case, so if there is a read maybe mapped between one read of the pair mapped here or the other mapped here, well, you insert a D here, and the insert length will get longer, and it'll become abnormal, and it'll reduce the score, right? So you have to consider all the situations in one step. It takes some time.
So when you like the randomized is completely randomized.
Yes, completely.
Because some of the reads, like the very reads, they give you some information on how the fragments are linked. Like there's always the green ones is just the anchor on the left, and the C fragments there, they are probably inverse.
Yes, I know. There is definitely some hint from the read we have, and also there's a very strong signal from the coverage.
So when you do the randomization, you might consider that.
We don't.
You can reduce the model space. You know that.
Yes, that's a very good idea. Thank you, but doing that usually costs more trouble. than the increase of efficiency, because like if you want to do a inversion here on C, like also you have decided like you whether you invert it or you copy and invert it later, and you have to decide like where you want to insert it. So like by doing the randomized method, you will reach the structure anyway.
But you probably tell like when you do the randomization, if you come up with a model, you can deny the model because there's reduce like computation.
Yes. Yeah, I can. So also we didn't take the hint from the coverage. Anyway, for this randomized approach, it is just totally randomized. We don't take any assumptions or anything like that. We didn't get the best structure. Yeah, that's another problem we have. So in most cases, so when we try to cluster these breakpoints together, we try to control the number of breakpoints clustered in one group. So usually, if the number of breakpoints is less than 8, we can definitely find the best structure in 100,000 iteration. We try that many times, and we think that's good enough for us. So the key point here is just we control the number of breakpoints in a cluster. But if there's some situation like the region is really too messy, you have like 10 or 20 different breakpoints there, we will maybe set a larger number of iterations there to run.
So this would not necessarily be a great tool for mapping something like the Prader-Willi-Angelman region, which is incredibly complex. Even in the reference sequence, there's a lot of nested and overlapping duplications and rearrangements within that region, even in the reference sequence.
I still think it works well. So like if you have a very messy region, but not all the blocks in the region are necessarily linked together. So maybe there are some part, like some local part, that all these regions are like all this rearrangement are happened within this block. And then you can solve this block first, and then expand it to a larger region. Like you can just solve this local region and fix it as the best structure you think is there, and then you continue add a additional breakpoint and try to solve it in the larger scale.
Okay.
But you are, of course, within these technologies, limited by technology and by the reference. So, I mean, there are going to be some reasons that no one's going to be able to apply with this technology just because you can't uniquely know where this group came from. For instance, this won't work with mobile elements, right? Because they're too repetitive. You can't really know if what you're seeing is mapped is correct. However, this is expandable to larger insert libraries such as mate pair or jumping libraries that are 3 KB, which do allow for amount of coverage to resolve some of the types of problems.
Okay.
So have you kind of benchmarked your performance as well as your quality of your output against the other 3 methods that you?
Yeah. So far we've just tested the performance of our breakpoint color, which is far back. So of all these breakpoints we could define within our method, we could detect, like compared to the the already defined simple deletions, we could find like around 70 or 80% of that breakpoint. So within the file itself, they have like 10% false positive. So in total, we may be able to get like 80 or 90% accuracy in defining the breakpoint. Well, for the later step, like for the iteration step, so far we've tried this on the, I shouldn't have done this. We've tried the iterations on the families, on the pedigree. Sorry. On this pedigree. And we have 3 cases on chromosome 2, 3, and 7. So this is a case on chromosome 2. We haven't validated it in the wet lab. but we think, like, look at the structure. It follows the Mendelian rules, and also we try to verify this with the PacBio data we have. And for this case, we already look at the PacBio data, and the PacBio data first support all the breakpoints we have, and also support this structure. Yeah, so for the next step, we will like send the samples to the wet lab and to validate all these structures in the sample first.
A quick note about the breakpoints too, is that 80-90% is based off of a gold standard set that was derived from many algorithms and validation sets. So compared to other algorithms on that same set, that's actually pretty good. It's not like 80% of a single algorithm. It's aggregation of multiple data sets put together this gold standard set of breakpoints. So it's actually pretty good relative to that. But this was like the highly validated set. Nobody even found the genome. Yeah, we single sequence a lot of breakpoints.
Why do you think 10 to 20% of this? Do you have any what problems?
The 10 to 20%. So yeah, I actually go back to check. So we got limited first, like if the region's too messy, we have too many signals here, we're gonna be able to tell it. For example, if a breakpoint is very close to the centromere or close to the telomere, or if there's like a tandem duplication like the satellite region. So I mean, the read are gonna map back very messy. So you have maybe a lot of the clip reads. The clip reads are everywhere. But if we define a breakpoint, we require maybe at least 5 reads to be clipped at the same position, which is impossible for this region, so we just miss that. This is one reason, and the other is we set some arbitrary parameters in the algorithm when we are looking for breakpoint. For example, if a breakpoint is defined by a cluster of aberrant reads, we require the cluster to be no smaller than consisting of 5 reads. And we actually did these like by setting different parameters like 5 or 10 or 15 or 20, and we try to fit the ROC curve, and we just pick the best the best parameter with like the best false positive and false negative. But it didn't cover all the breakpoints we get. Also, like by defining the breakpoint, we didn't consider the coverage. So if there is a very strict coverage change there, it could also indicate some breakpoint there. But because adding the coverage redepth calculator is gonna take the 2-hour run to 2 days, so we just get rid of that, and we sacrifice a very small proportion of the breakpoint we can define.
So what tools did you use to do?
Python, just Python. So I write all this scripts myself.
You gonna make it available to people? Or?
I'm trying to. Maybe this summer.
Any more questions? Okay, thank you again. Very nice.
Thank you.